In the central dogma, DNA is used to make RNA, and RNA is used to make a protein. So if you follow that pathway, if you change the DNA, you change the protein. Now, epigenetics is the study of the expression of genes uh, in our gene pool and what causes changes in how genes are expressed. So, every cell has two sets of DNA. You get one from mom and one from dad. The cells that are produced after fertilization differentiate. They become all the different parts of your body. They do this by highlighting or activating certain parts of the DNA in each type of cell. So, pancreatic cells will make insulin, and bone cells will pull calcium out and stockpile calcium inside of them to make rigid bone structure or matrix. Well, there are factors that can turn on or off certain parts or sections of DNA inside of a cell. There are two different chemical reactivities that are identified here. This is acetylation and methylation. Acetylation, when the DNA is acetylated, it keeps the histones loosely coiled, and this loose coiling shown in the diagram B down here, this loose coiling makes it possible for the DNA transcription factor, the protein that's responsible for transcribing the DNA, can fit in here and grab onto this DNA. So the only way we can get copies of a particular gene to make a protein is if we're able to get the protein in here, the enzyme, to get it in here and copy this DNA. We call that switched on. This is active open chromatin, all right? And the histones are loose. That is in contrast to methylation, and methylation tightens and locks down the histones. And this is called silencing a gene. Silencing a gene causes this methylation almost wraps around. The DNA is attracted more tightly to the histone proteins, and so it won't unravel and won't unwind. So it prevents the DNA transcription factors from accessing the actual DNA. I'd like you to pause at this moment, just take a time out, because this concept is highlighted really nicely in an epigenetics crash course. It's I know it's 9 minutes, and you've heard me say before that he talks kind of fast, but this epigenetics crash crash course is worth the time, because they go into a lot of great graphical representations of how it works. There are several different reasons why genes can be turned on or off. You can have chemical exposure, either in the case of hormones, drugs, pollutants in the environment around us. We see plenty of examples of chemical exposure in the form of cancers, like leukemia or lung cancer. Exposure to chemicals can mutate the DNA and cause cells to start to replicate out of control. That is what cancer is. There are environmental conditions that can influence the expression of a gene, like in the rabbit shown down here below. This rabbit has white hair on its body and black hair on its extremities. An experiment was done where the back of the rabbit was shaved and an ice pack was strapped on. And as that hair grew back, it grew back black. So, environment is included in the list of factors that can influence the expression of a gene. So, the rabbit has black fur on cold areas of its body. So its ears are cold, its nose is cold, its feet are cold. Its back wasn't cold until we put the ice pack on there. And that switched on a gene. That environmental influence switched on a gene for dark fur because in nature, dark objects warm up more when exposed to sunlight. It absorbs ultraviolet and infrared radiation a little bit better so that it retains heat. Third on my list are the oncoviruses. Oncoviruses are cancer causing viruses. They are known to cause mutations in human DNA. The human papillomavirus or HPV is an oncovirus. You may have received the Gardasil vaccination to prevent the infection of HPV in your body. There are also human intervention can cause mutations. We know that we can cause mutations in organisms. We can deliberately change the genetic material. We can do this randomly by using radiation. Uh, we know that nuclear radiation, and this includes radiation from the sun because the sun is really just a nuclear furnace. We can use radiation to cause mutations. We can use other viruses that are specifically designed to cause mutations. We can even influence the expression of genes by doing a selective breeding program that can change which genes are more often expressed because we can control which ones are passed on from generation to generation. Which brings me to, because we're talking about human interventions, we're talking about genetic engineering now. We can use technology, our knowledge, to alter the genetic instructions in an organism. This is a type of biotech that is very useful, and I say very useful in a novel method, meaning it can be, we can make pets like neon tetras. It's also great for treating disease. We use transgenic organisms, which are organisms that have genes that are not found in those organisms. We use transgenic organisms to do genetic research all the time. It's commonplace now, and they contain DNA that has been recombined with organisms' DNA that doesn't belong. We also do selective breeding, which I talked about earlier, where breeders or farmers choose individuals with the desirable traits and only breed those in an effort to generate more of the same type that they want. This is the oldest form of genetically engineering that humans have ever done. So people who say, "Oh, we shouldn't be messing with genes," well, we've been doing it for a long, long time. Dairy cows are bred to produce as much milk as possible, uh, more than they ever would in nature. The corn on the cob that you eat in the summer is a result of uh, thousands of years of manipulating and selecting grains to grow the way that we want them to. Uh, the original corn plant did not produce a huge cob with giant seeds that were edible like that. It was more like a grain like wheat. And even wheat, we've selected to produce the type of wheat seeds that we want so we can use it to make food. So, in general, selective breeding is very old, very common, and we do it even today with our dogs and cats and all sorts of pets that we like. There are pros and cons. Inbreeding of offspring, meaning you only breed individuals with the same traits so you can maintain a characteristic. Well, it's more likely that those individuals, when they're inbred like that, are going to have some recessive trait that might be expressed. This happened with hip dysplasia in lots of larger breed dogs like labs or German shepherds, Rottweilers, uh, golden retrievers. Uh, they all have a risk for hip dysplasia. In humans, the Russian royal family has a genetic problem of hemophilia. Historically, if we look back at the family tree for the Russian royal family, there was a lack of royalty available to breed with, and I say breed in the most noble of ways, of course. The Russian royal family was only allowed to marry and have families with other royalty. And if there are limited numbers of royal families available, then sometimes your cousins and second cousins will just have to do. And so what happened was there is a recessive characteristic, a recessive gene lurking in the Russian royal family, hemophilia. And because they were breeding or mating with individuals from the same family tree, they also had the recessive hemophilia trait found in their gene pool, and more kids were being born with that particular trait. Since I'm talking about the Russian royal family, I decided I'd Google it and see if I could find a pedigree, because this is one of those instances where we can use a pedigree to follow a trait through a family and its expression. And it turns out it's not just the Russians who had the problem. In fact, it was Queen Victoria of England who was the original carrier, and Queen Victoria had several children here that we could identify: one, two, three, four, five, six, seven, 8 children, 3 of whom were carriers or expressed hemophilia. Now, I mentioned that the Russian royal family, and the reason why I mentioned the Russian royal family is because uh, Queen Victoria of England had one daughter who married and had one son. That son married their cousin. This is a cousin right here. This is her sister's child married the son here and had 3 children. All right, so this is the Russian royal family here. These are, it looks like, Spanish royalty over here. But because the English trait was transmitted into the Russian family and into the Spanish family, I could make that argument for both, but it's the Russian family that was doing the inbreeding anyway. But anyway, that's all I was going to say about that, just so you had a chance to see it and we can see how we can use a pedigree chart to track traits in a. Tay-Sachs is a genetic condition that's frequent in Jewish populations, and so frequently uh, Jewish families of common descent, which is a lot of them, uh, the Jewish families will be tested for this gene before they have kids so they can analyze their risk. We use gene splicing to put genes into an organism that don't belong. This is how we do our genetic research typically. We use the restriction enzymes that we talked about earlier in our first uh, genetics video, where we used restriction enzymes to do a DNA fingerprint, the gel electrophoresis. Now we're going to use restriction enzymes to cut the DNA and allow a splice to be put in. And we can splice into a bacteria a gene that doesn't belong, like a human gene for insulin or growth hormone or protein, whatever human protein we want to make, we can have bacteria do it because bacteria have this blue ring here is called a bacterial plasmid. This plasmid is just their ring of DNA. So we can cut this plasmid with the specific restriction enzyme like EchoRI or BAM or Hindi. These are all names of restriction enzymes, and they cut at certain A, T, G, or C sequences. And we can input a gene that we want and cause that bacteria to produce a protein that wouldn't normally be there. This is a way we can treat disease. If you have a person who has to take insulin in your family, the insulin they take is from genetically modified organisms, bacteria. They're transgenic bacteria that have human genes in them, and they produce the insulin that your family member needs. This is the same with just about any hormone that you have to take for a condition you might have. If you're taking a hormone supplement, if you're using birth control, that hormone is produced this way. Here's some examples of some of the first transgenic mammals and the first transgenic organisms that we produced. Um, this particular picture on the top left is a tobacco plant that has been genetically modified to have the firefly luciferase gene inserted into it. So it actually glows. This picture is a picture of some genetically modified fish that have had the luciferase gene inserted. Excuse me. We have genetically modified mammals as well. Here are some cute little kittens that will also glow in the dark. I want one. That way you never have to worry about wondering if the cat is sneaking up on you in the dark. But this is a monkey that has had that same gene inserted. All right, this is a fluorescing protein. It's called GFP, or green fluorescent protein. That is the gene that's been inserted into these mammals here. All right, now let's talk about some actual therapeutic uses. For genes and genetic engineering. An absent or faulty gene can be replaced by a normal working gene in a lab. We can now take cell samples out and we can modify the DNA of those cells in a lab, grow them in a lab culture, and then put them back into the body corrected. This is done using a virus to deliver the DNA. Viruses do what viruses do, they infect cells. And if we can program the virus to infect cells with DNA that is desirable, then we can utilize the activity of those viruses to deliver DNA that is missing. So we can have this hemoglobin gene inserted into a cell, and now that bone marrow will produce the hemoglobin that's needed. Okay, so this is our latest and greatest technology we use for genetic engineering. This is a process called using genetic scissors, or the CRISPR-Cas9 system. This is a system that was found in bacteria several years ago now, and bacteria use this particular system to identify viruses that are trying to infect the bacteria and kill the viruses before they can actually cause the bacteria to die. Well, the bacteria has developed a system to read genetic material and cut it at very, very specific locations. And we stole this protein system, the CRISPR-Cas9 system, and we use it now to edit DNA. There are two fantastic links provided uh, in your slideshow that you can actually click on and you can watch. I recommend you take the time to check it out now before we continue, because I just cannot do this justice. The technology exists now to be able to manipulate human or any DNA in any way we want, to insert any gene we want, just as easy as getting cells into a lab and setting them up with this CRISPR-Cas9 system. We can generate what's called a guide RNA. This is the green section here. This guide RNA will look for a specific sequence in the DNA, a matching sequence. So all we need to do is know a code that we want to find. So we can actually create the guide RNA in a lab, and we generate that guide RNA to match a code that we're trying to find in a person's body. And as soon as it finds that code, it will cut the DNA. Now, normally, if uh, if this were in a bacteria cell, it would just cut the virus DNA and it would mutate it and cause it to no longer function. We can do this with human genes. We can just cut it and disable that particular gene. It gets our bodies have a reassembly protein that will put that back together, but it patches it, and the patch causes a mutation. And the patch prevents the protein from being made the way it was before. So it silences, essentially. We're using the term silence, and it may not necessarily mean exactly what you think, but it turns off that gene. The protein is no longer functional. Now, that's a way to stop a faulty gene from working. But we also have the ability to cut out the section and insert the correct material or a different gene. So we can identify the target gene, or we call it, there's a PAM sequence, which is a section of DNA that is recognizable. The guide RNA will match that target gene, and the CRISPR protein will cut the target where it matches. And then you can either deactivate it, which means it's off, which I was just referring to, or you can have a different sequence inserted to correct the gene. So if you don't produce a protein properly, the CRISPR-Cas9 system can actually correct it. But here's the catch. You have to do this early on or have a very specific region that the cells need to be affected in, because we can do this and correct cells at the embryonic level, assuming you have a fertilized egg that is only a few cells now. It's dividing, it's dividing. Right now, the cell numbers at that stage are feasible. You can correct the DNA and have the organism, your child, develop with the characteristic corrected in its whole body. But you and I, we are fully developed. All right. Our organ systems are organized. All the genes have been turned on or off that are going to be, like over time. If I have a condition that I need to correct, I can't correct it throughout my whole body. I have to target certain cells. And this is one of the challenges that scientists have been trying to overcome. It's the targeting that's the hardest part. You have to find a way to get that CRISPR-Cas9 system into the cells that need it to fix the condition that exists. They used this particular system to correct a vision impairment in a woman, where they just injected into the eye the CRISPR-Cas9 system proteins, and the CRISPR-Cas9 guide RNA was created to correct a gene in the cells of her retina. And it actually was able, over the course of time, to correct her vision. Her not like it wasn't like a correct your vision so she didn't have to wear glasses. It was the ability to correct her blindness. She was blinded by a degenerative retinal disease, and they identified the gene, they programmed the correct version, and they inserted it into her eyes, and it corrected her vision. She gained her vision back. So this brings me to a hot topic, which is cloning. Cloning has been going on for a long time. When I say long time, I mean the first mammalian clone was Dolly the sheep. And Dolly was born in 1996. 1996 was the first mammalian clone produced. We have not yet produced a human clone, but we do this by a process called nuclear fusion. So we take a donor egg and we remove the nucleus, and we insert the nucleus of a cell that we want to clone. So you take the cloned person's DNA and you put that cloned person's DNA into the cell. It undergoes a mild electrical current. They shock it. It'll trick the egg into thinking that it was just fertilized, and it will start to divide. You then have to implant those fertilized cells, the embryo, into a surrogate mother who will then give birth to a clone of the donor. Not the egg donor. The egg donor's DNA has been removed. It's the person you're cloning who provided the sample. Their DNA was put into the egg, and now their DNA is the DNA that is in the cloned cells. The baby will be a clone of that individual and uh, will have the exact same DNA code, but not have life experiences, education. There are some epigenetic factors that can still be transferred, but they are cloned. Population genetics is an interesting topic, and we start talking. This is on the edge of evolutionary activity here. But when we start talking about the gene pool, this is the total of all genes present in a population. So if you're thinking of your classroom, you might say, oh, how many blondes are there? How many redheads are there? How many blue eyed people? This is the gene pool. What are your options? And the gene frequency is how often that gene occurs. This is the percentage of individuals in a population that has a gene. Well, there were two statistical geneticists that developed this proof of evolution. It's called the Hardy-Weinberg principle. And under certain conditions, they said the gene pool of a population will always remain stable. All right. Now, it will only remain stable if and only if the following conditions are met. One, you must have a really large population, which for endangered species isn't the case. All right. Like if you think of a species like the cheetah or the panda, these are small populations. Right. But in order for evolution to not occur, all right, you've got to have a large population. You can't have anybody migrating in and out of the population. You've got to have a stable group that does not leave. So if you were thinking of your classroom and you were to say, oh, all the redheads were leaving the classroom, or everyone with blue eyes is out, right? That can't happen. Mutations can't occur, and organisms in the population have to mate randomly. Now, the reality is none of those criteria are going to be met frequently, all right, or at least all at once. So the result is changes in a population over time that results in the evolution of new species. Mutations are the source of those variations. So we know that environment can cause mutations. We know that humans have the ability to cause mutations. It's those variations that create new combinations that lead to desirable traits, either environmentally desirable, like you're just better able to survive. You can hunt better, you can see better in low light conditions, so you can catch your food and you survive. If you have the traits that make you more survivable, it increases your survival rate, then your population, your gene will increase in frequency because you're going to be able to pass it on. There's a great crash course video, whoops, as I just cover it up. There's a great crash course video on population genetics that you should take the time to look at and evaluate, because we see over time changes in populations that we recognize within human lifespan. Right. We have historically suggested that evolution only takes place over thousands of years. Well, we have seen evolution in the short term. We call it microevolution of populations in the short term. There's some really great evolutionary genetics that we're going to be sharing with you in our unit on evolution.